Okay. Are you working? Yep. I'm gonna go check on my phone.
So which one am I supposed to actually present on? Okay. I think this is it, right? Is this the right screen? And I can hear myself, so okay. Should I be hearing myself in here, or is it through there? Okay. Okay, if you don't mind, I'm going to try going on and try to finish off as much of today's lecture as possible, but also spend a few minutes at the end talking to you about your literature review and things that you might want to try to do for your literature review as well. So our current model that still exists to this day in 2017, this model, by the way, is about 17, 18 years old. I know, you can't get any more excited about LTP than that, right? Awesome. So this current model suggests that this change in the size or the amplitude of LTP as a basis for learning and memory. So I showed you that with Giesi, who was part of the Alcino group, that they made this correlation between LTP, learning and memory, and that CAM kinase 2 mutant. And we think that this is actually due to either a change in the conductance or how many ions flow through the AMPA receptor, or the number of AMPA receptors that are found after stimulation through LTP. Much of this was actually shown by Roberto Malinau to also have occurred in the mid to late 1990s, where he tagged GFP onto one of the subunits of the AMPA receptor known as GluA1, and what he showed was that in a CAM kinase 2 dependent way, after high frequency stimulation, he observed the movement of GluA1 containing GFP into the synapses only after he had high frequency stimulation. And this was the work of Roberto Malinau. And this set off a whole new field of investigation trying to identify how did these receptors actually move into the membrane, why they move, etc. And then LTD was rethought as the removal of different AMPA receptors. Still AMPA, by the way. AMPA is the receptor that's moving. NMDA, still in 2017, has not been shown to move out of the synapse one way or the other, whether away from the synapse or toward the synapse. And this LTD is also thought to be a resetting of synapse. We're reworking this model. We're not 100% sure if this is correct now, sort of in the tail end of 2017. And some people are actually rethinking the importance of LTD. Some individuals, for example, in the US, think that LTD is the process that is more important and it should be the dominant mechanism that underlies learning and memory, not LTP. So we'll have to wait to see how that particular train of thought actually works out. So this is a slide that I want you to kind of understand some of the basis for. I've gone through some of the mechanisms with you today, and I'd like you to make sure that you understand the overall picture. If this is a synapse that is either getting stronger or getting weaker after high frequency stimulation, what we're really talking about is the movement of these receptors here, which are the AMPA receptors. The induction and expression of LTP begins through the NMDA receptors. And again, funny enough, the induction and expression of LTD begins with NMDA receptors too. We still need NMDA for LTD as well. We still need calcium to flow through NMDA receptors. The molecules are just a little bit different to cause LTD. So ultimately here we're going to be activating molecules like CAM kinase 2, the work that Alcino Silva and others did. And much later on, people here in Toronto, and I was part of the group that actually was involved in trying to figure out the mechanisms, the molecular mechanisms by which the AMPA receptors actually made it up to the cell surface. There was actually a really big race, and I remember this was such an exciting time to be in the lab. It's one of the few times where I actually really liked sleeping in the lab, just because we wanted the data out as quickly as we could. We knew that there was a group down at Harvard working on exactly the same thing, and we knew that it was going to be a race to get our data out either before or at the same time as they did, because no one knew what was actually happening in LTP. We thought we had a pretty good idea that it was the movement of AMPA receptors up to the cell surface, and we were working in Yutian Wang's lab at SickKids to try to be the first group to actually publish this. It was a tie. We tied with the group down at Harvard with Morgan Sheng and his group to actually show at exactly the same time that LTP was the insertion of AMPA and LTD was the removal of AMPA receptors from the cell surface. And again, it's sort of withstood this test of time that kinases are responsible for the insertion and stabilization, and phosphatases like PP2B or protein phosphatase 2B or protein phosphatase 1 or this other phosphatase, which is off to the side known as calcineurin, dephosphorylate AMPA receptors or cause them to be endocytosed. So there's a very good symmetry between insertion and strengthening and the removal through endocytosis and removal causing LTD that we think is still really important to this day. This is a mechanism, both LTD and LTP, that I'd like you to be familiar with for the final exam. I will be giving you some practice short answer questions as part of your participation related to manipulating or to demonstrating that you understand these processes. And again, we'll do the same thing that we did prior to the term test, where you'll be posting your answers anonymously on the discussion board. So I hope that I've tried to demonstrate that there's a good connection or a strong connection between the processes of AMPA receptor movement and LTP, and that there might be a strong connection between LTP and learning and memory. Alcino Silva's group showed that impaired LTP was associated with impaired learning, and I showed you that earlier as part of those graphs. Showing the escape times were very different between the mutant mouse and the wild-type mouse. Once we were at this stage in the early 2000s, it became another matter altogether to try to show that LTP is equal to learning and memory. Think a little bit about that. Does strengthening of synapses really cause you to remember better? If that's true, how are you going to prove to me that LTP fundamentally makes you learn better or to remember better? It may sound really trivial, but this was not trivial. This is actually one of the hardest things in neuroscience to prove. Now, again, I can ask you, can you demonstrate in any way to me, using any paper that you've read, outside of the ones that I'm going to present to you, that after you've learned something, that something has changed in your brain? Any papers? So you're not going to tell me about the London taxi drivers and their hippocampal maps and the increased size of the hippocampus? Yes? Okay, so tell me a little bit about that study. No? Nothing about that study? Yes.
So I think they monitored taxi drivers at the beginning of their training and at the end, and it was like a really extensive training to learn all the roads. And by the end of their training, if they had succeeded, their hippocampuses had grown in size.
Okay, so again, most of you will kind of have the Reader's Digest version where these taxi drivers in London, they actually have to go through an accreditation process that takes several years. They can't get their license right away. They have to demonstrate and practice and learn all the different streets that are in London, a very big city that has a lot of different streets, because it involves the hippocampus that's involved in spatial navigation. When they look before and after, their hippocampi got bigger. What does that actually mean? Their hippocampus is bigger. What does that actually mean, though? Like, can we see bigger synapses like on an fMRI or on a structural MRI? So when you say bigger hippocampus, what does that actually mean? Did I grow more neurons? Possible. We talked about neurogenesis in the hippocampus. Maybe that's possible. But what is the reality if you go back through those papers? What really happened to their hippocampi? And it did get bigger. Like, structurally, it was bigger. Did the neurons get bigger? Were there more neurons? When you go through that study, what did they actually show?
Arborization.
Maybe arborization, but more importantly, when you're looking at this, and we're going to come back to this and talk about this in the very last lecture, where we're looking at how do you actually prove and demonstrate a phenomenon, and you're looking at what happened structurally. One of the things that they found was that the size of the hippocampus was bigger mainly because there was increases in the amount of myelination in some of the different neurons there. They were sending out their information faster than they were before. And we're going to see exactly the same process where people previously saw that there was a thickening of the cortex following meditation and mindfulness practices, but we now know that that thickening isn't due to more neurons or better arborization. We now know that it's due to better myelination, and somehow this glial process and sending out information is more important. So we have all of those different issues. Now we're going back to this issue that I need you to be able to prove to me on the final exam that you understand how to link LTP with learning and memory. So Alcino Silva's group back in 1997, 98, and 2001, they tried their best, and the technology that was available at the time, they tried their best. And we could certainly talk about watching molecules moving into synapses. We could track different AMPA receptors, but that's not equivalent to showing me that as you are learning something for the very first time, that your synapses are getting stronger. That is fundamentally different. I need you to be able to show me in real time that as you're learning something for the very first time, that you're generating LTP, you're strengthening your synapses. And that has been incredibly difficult, and this direct evidence is actually almost equivalent to winning a Nobel Prize. There's very little evidence that's directly correlational to this that actually gives us the proof that shows that LTP equals learning and memory. As you learn something, if I were to stick an electrode into your brain as you learned, would I actually see some of your synapses going and increasing in their amplitude of response? And this is really challenging because over 60 years, if you're looking at an intact animal, finding the right group of synapses that as an animal is learning, finding the exact number of synapses, incredibly difficult, right? So each single neuron is making 1,000 to 10,000 synapses with other neurons. How am I going to make sure that I'm tagging the right one of these? There's also a lot of variability across the hippocampus. So LTP is incredibly difficult to detect unless I have it in a slice in a controlled condition. Not every single synapse, as I mentioned, in the hippocampus at any given time is going to be strengthened. So think about what you're doing right now in this room. You're sitting here trying to pay attention to what I'm telling you up here, but you have all sorts of other information that's bombarding your hippocampus through that entorhinal cortex at the same time. The vast majority of that is never going to be strengthened. You're never going to get LTP. So how do I know which synapses are actually allowing you to learn what's up here right now? So there's also this whole issue, which we're not going to address today. If you take a fourth-year course down the road, you might address this. Is LTD actually more important than LTP? So is the deconstruction of a synapse more important to LTP overall in terms of learning? And maybe that's really the mechanism that we should have looked at. So we don't know the best way to do this, and there are only, to this day, so I continually scan the literature, this study was published and was a landmark study that's been cited in almost every neuroscience textbook, and I'm going to take you through some of their data. But this landmark study was actually one of the first. But if you look overall, can you prove now in 2017 that LTP occurs and then that results in learning and memory? There are only 24 papers in all of existence that actually can demonstrate LTP as learning and memory are being formed. And again, this is a fairly old paper now, so think about the length of time that has occurred since this paper back in 2006. Do any of you know this individual here or this individual here? I kind of doubt that you would, but hey, you never know, gotta ask. So this is Jonathan Whitlock, and some of you might know Mark Baer, who is one of the authors of a very famous neuroscience textbook. And these were actually the first individuals in 2006 to demonstrate for the very, very first time that learning actually induces long-term potentiation in the structure that we've been looking at within the hippocampus. They were one of the first groups to make that link that people had found back in the 2000s that AMPA receptors, LTP, might underlie learning and memory. And he needed a very good learning model, and he needed some sophisticated technical analyses for him to be able to prove this to the scientific world. It was not a trivial feat. This is, again, still withstood the test of time back since the year 2006. No one has been able to refute his data, and it's a pretty remarkable finding. So when you're looking for a model of learning and memory, and you're looking for learning and memory, and you want to see LTP in real time, you need to have a really good model for learning and memory that everyone is going to be able to agree on. It has to be a simple model. And I want you to understand this model. And what they did was they used a model where you had to learn something. And this was a form of inhibitory or passive avoidance. Many of you will know, because you've taken psychology courses, that animals generally like being in the dark, especially mice as well as rats. They're not normally acclimatized to being out in the open in full broad daylight. So they like dark places. That's their normal environment. You have to teach them, they have to learn, in other words, to avoid being in the normal environment. They like going into the dark. You have to teach them, they have to learn not to go into the dark. They have to learn to avoid their normal response. So there's a form of learning. It's very quick. You can see it right away, and you need to be able to, again, buy into this being a good form of learning. So this is what they're using. They're using a two-chamber system that I'm going to show you in the next diagram. There is a lighted chamber. The mouse, or rat in this case, doesn't like being out there exposed in the light. Its normal response is, when it's up there, is to step down. And below that chamber, there's a darkened chamber. So normally they'd want to step down, but you are teaching them, and they are learning, that when you step down, you're going to get a little foot shock. So if they learn that really well, they will learn to stay on the lighted platform. Does that make sense to everyone? So this is their learning model. This is that learning model that I was showing you earlier. So they have to learn this. This is not their normal response. They have to learn this because it's counter to their normal nature. So this is a form of learning. This is also experience driven. The more often they learn that they're going to get a foot shock, the faster that they're going to learn not to step down. So it's also activity or experience driven. And it also results in memories being formed. If you take them away from this particular model for a while, and then you put them back on that platform 1 week later, will they still remember that 1 week later? That's another critical test, and they will. They will remember this for a long period of time. So again, this is their model, this passive or inhibitory avoidance learning, and they want to know, is LTP involved in this learning process? Can they see LTP as this animal is learning not to step down into this darkened chamber? So I'm hoping that's fairly clear for you. So this is what I'm looking at here on the right. So apologies on this. There are two different models for passive avoidance. What you should be looking at is the model here on the right, which is this step-down passive avoidance model. So their normal response when they're up here in the light is to step down into this darkened chamber. Rodents in general like to be in the dark, and as a result, when they do so, they get a shock. They learn very quickly, if they are able to learn, to stay up here on this lighted chamber up here. Does LTP occur as they are learning not to step down? That's the key point here. Can anyone demonstrate that LTP is occurring? The problem is the same situation that I've just outlined for you before. The hippocampus, it may seem like a small structure to you, but overall, like, there are 2 million neurons that are packed together in this structure. How do I know out of that 2 million neurons that I'm actually recording from the right one while this animal is learning? It's a stochastic process. It's a probability process. So again, think a little bit about this problem. How am I going to show you that as it's learned this and as it's learning not to step down, that LTP is actually occurring? Will I be able to demonstrate this strengthening? So before he goes into doing all of this, he actually does a number of molecular studies, and this is from his 2006 paper. I'm going to take you through parts of this, but I don't need you to memorize all of the data. I don't need you to memorize the overall size effects. I don't need you to memorize all of the different graphs. The important things are what I'm going to be outlining for you over on the bullet points, and I hope that some of this makes sense. So one of the things that they're going to be doing is to be looking at one of the subunits for the AMPA receptor. Remember, the AMPA receptor is the one that in the early 2000s was hypothesized to move around, to be phosphorylated by CAM kinase 2, etc. And one of the things, again, if learning has occurred, one of the outputs, according to that model, before showing LTP or anything else, before doing any of those things, one of the things that he wanted to demonstrate really quickly is to show, is there phosphorylation through a CAM kinase 2 dependent mechanism of this AMPA receptor that everyone else in the world has shown previously. And what I want you to know and pay particular attention to is this bar here. As the animals are trained and as they learn, one of the things that he finds that, yes, indeed, on the GLU-R1, which is equivalent to GLU-A1, the old nomenclature in 2006 was GLU-R1. That same part of the AMPA receptor is now called GLU-A1 in 2017. So I'll use those terms interchangeably. You can also use those terms interchangeably on your term test or your final exam. So GLU-A1 or GLU-R1, what he demonstrates is as the animals learn, after he takes their brains out after learning, he does indeed show that the subunit is phosphorylated on this serine residue, serine 831. This is one of the rare times that I'm going to ask you to make sure that you know and memorize a number associated with a particular subunit. The reason for that is this. There are actually two serine residues on AMPA receptors. There is serine 845 and serine 831. 831 had been shown by 2006 to be the residue that after learning and memory to become phosphorylated, whereas the serine 845 had been shown by 2006 not to have been increased in phosphorylation. And he showed exactly that as this animal has learned not to step down into the dark, to stay in that lighted chamber, after this training, he sees this phosphorylation in this panel of only serine 831 of the GLU-R1 subunit. So again, can you think of a reason why? Oh, I actually haven't told you this, but CPP is another compound which is an antagonist for the NMDA receptor. Do you remember any other NMDA receptor antagonist that I mentioned to you earlier today? So MK1 is one. What's the other group? AP5 or APV. So again, this is an NMDA receptor dependent pathway. So if he does this training by injecting in CPP and then he measures, did the animals actually see increased phosphorylation? This is completely blocked with CPP. It is still NMDA receptor dependent. Learning, LTP, always has to be NMDA receptor dependent. We've seen that with Alcino Silva using AP5, and now we've seen this with Mark Baer's group using this CPP, which is an antagonist for the NMDA receptor. So again, without going into a lot of details, and I don't need you to go through all of this, they actually do see an overall increase in the levels of the AMPA receptor subunits, both GLU-R1 and GLU-R2. And this subunit here in panel F is the subunit for the NMDA receptor subunit. Interestingly, they see no change in NMDA. Is that surprising to you or not surprising to you? So we don't think that NMDA receptors change, and what he shows is that NMDA receptors don't change their overall protein expression levels either. So again, it's fairly good proof that his model seems to follow all of the different dynamics that we would expect for this LTP model. This is not showing the link between LTP and learning. It's showing that his model, the step-down model, has all the biochemical features of what we would expect in LTP. Is that clear for everyone? Okay, so again, this compound here, CPP, is one that you need to make sure that you know, and I'm hoping that you will remember this subunit. The serine 831 is the more important one for phosphorylation, especially as you go through question 3 on the online quiz. So if you were to take a look at this, so which one of the following would be increased in the hippocampus of a rat as it undergoes a successful spatial learning model? So which one of these would you see changed? So would you see any change in serine 845 if it is undergoing LTP? Would you see a change in the NR1 subunit levels or phosphorylation of NR1? Or would you see a change in the GLU-R1 serine 831? So I would agree with the majority of the class, and you'll see that it's a very similar type of question on your online quiz, that we really, if this works, should see a change in the phosphorylation levels at serine 831, which is what was shown here on a biochemical assay. We wouldn't see a change in NR1. We wouldn't see a change in serine 845 either, even though they are on the same subunit. Do you have any questions about anything before I go on? Or anything that you're not clear about? Anything that you'd like to double check on this particular quiz question? No? Okay, so I haven't yet shown you how all of this actually relates to each other. Um, at the time in 2006, there were only really two labs that were capable of doing this experiment. One was the lab, there were probably three. One was Mark Baer's lab, and Mark Baer's lab actually created a multi-electrode array that they put in to the brain of the hippocampus. So I told you that it's almost impossible to guess which one of these synapses, which one of these CA1 neurons is actually going to see LTP as this animal learns this task. Because there are 2 million of these. How do I know that I'm recording from the right one? So it would be a very difficult task. So I'd have to record from a huge number of animals. Mark Baer's solution to this was, I don't care, I'm Mark Baer. I'm just going to record from every last single possible neuron that I can at the same time. So he created this multi-electrode array. So he recorded from, at the time it was remarkable, now it's like pretty commonplace. So he recorded from like over 24, sometimes 36 different neurons at the same time in a single experiment. By doing this, from recording from a huge population, again, I use the word huge, we wouldn't consider that big anymore, but by recording from this huge population, one of the things that he was able to show very definitively, biochemically, he showed all of the hallmarks for LTP. And here, what he was able to show in an animal intact, so this is not where you've taken the brain out, this is an electrode in the animal, you are recording in real time as the animal is actually learning not to step down. One of the things remarkably that he was able to show in a living animal in real time was that some of these synapses actually got stronger as they learned this task. As they learned that, wow, just got a foot shock, I shouldn't step down, these animals actually learned. Some of these synapses got stronger. He also saw, exactly as predicted, many of the synapses that he was recording from actually had no effect. Now, you might argue, well, it's just a random effect, right? It's just random. So he just got that by chance. Like some of these synapses are going to get stronger, some are not going to get stronger. So yeah, sure, the ones in red got increased after they learned not to step down. But that's going to happen anyway. It's just a stochastic, probability-related effect. But when he does the control, where he is just allowing the animal to stand on the platform, not learn, he never sees strengthening. So he only sees this in a learning paradigm. So this is really related to seeing LTP, as you see in these red bars, or in these red blotches. This is from a single group of neurons, and these other colors are from other groups of neurons. They're not being strengthened. He's actually able to demonstrate LTP is occurring in real time as this animal is actually learning. This was one of the first ever papers to demonstrate and make that link between LTP and learning in an intact animal model. Again, I'm not sure if you are amazed by that. I don't think now, in terms of the technology, that we would be amazed, because this paper just came out this week showing that this silicon electrode that is going to be used in human studies can actually record from several hundred neurons at the same time when implanted in the brain. So that technology at the time, where we're recording from like 24 to 36 neurons, was remarkable, but we're now able to record from well over 100 off of just this one silicon electrode. And if you have multiples of these, you're going to be recording from like up to 1,000 different neurons in a region at a time. So our technology is always advancing, and especially in neuroscience, we're always trying to figure out better ways to answer these remaining questions that we have. So is this model correct? That's the big question. We don't know. We think this model is correct. There's a lot of evidence to suggest that this model is correct. Most of it has withstood the test of time. People are kind of questioning, is LTP really the model that is important or not? One last thing before I switch over to sort of a different study. How many of you like Mark Baer's model? Well, it's Mark Baer. Of course you're going to like his model, right? How many of you don't like his model? If you don't like his model, why don't you like his model? So, how many of you think that a foot shock is a stressful event? The, the rat stepping down gets a little foot shock, stressful. Do you think it's also a fearful effect? Every class that you've taken in undergrad where you have learned about fear memory and remembering that fearful memory, what area of the brain has that been implicated with? Is it the hippocampus? It's the amygdala. And so when this paper came out, Mark Baer was actually criticized. Like, if you can criticize Mark Baer, go for it. But they criticized him like, wow, but really you're looking at fearful memory. And one of the things that you should be recording from for LTP is actually the amygdala. Why are you recording in the hippocampus? But again, he's Mark Baer. So he says, well, is this platform a position in space that the animal is aware of? And of course, people say yes. And so are they actually learning something that's hippocampal dependent? Which of course is also a yes. So the take-home message is, if you're a big enough neuroscientist, you can always find a way to answer all of your critics. And he answered it with a resounding, very logical solution that many people were kind of wondering about. And again, it has withstood the test of time. So again, we're looking at this trafficking of AMPA receptors, and we think that it involves the NMDA receptors. We think that it involves CAMP kinase 2. It involves having more exocytosis or insertion of these AMPA receptors for LTP. And we think that the removal of AMPA receptors is also dependent on phosphatases that dephosphorylate AMPA receptors and move them into the cell, making it less responsive. So there are less AMPA receptors on here after LTD. Also, though, NMDA receptor dependent. We're not 100% sure if this also occurs at inhibitory GABAergic synapses. That's a big remaining question. Many people believe that yes, it's true, but inhibitory synapses don't look at all like these excitatory synapses. These inhibitory synapses are broad and flat. They're not found in the same places. They would be found, for example, down here. There are completely different groups of proteins down here on the dendritic shafts rather than on the dendritic spines. So we're not 100% sure yet. There's a lot of work that remain for you to maybe try to figure out. So the last thing I'm going to end this particular group of slides with is to just take a look really quickly to reinforce some of these concepts with a slightly different study. We've looked at this, and you don't have to memorize anything from this slide. We've seen this slide before. You know about the stress pathway. You know that stress is a very big impairment to your learning and memory, right? I've heard all of you talking about, I wish I could just write this test without being stressed out, right? And I think that's one of those things that we think a lot about. And we now have the tools to be able to see, is stress actually an impairment to memory? And how would we go about looking at whether or not some sort of psychological stress, not a physical stressor, how that might cause the release of molecules like glucocorticoids that might impact the brain and might affect your learning and memory? Because the one area of your brain that has a very, very high concentration and density of glucocorticoid receptors is your hippocampus. In fact, that's one of the areas that lights up the most when you look at glucocorticoid receptor expression. It is very vulnerable, in other words, to stress. You can't remember, you can't recall things when you're incredibly stressed out. So this begs the question, which we often talk about in the office, right? So we're often interested in, how do we maximize the learning that our students are getting out of these different courses? And how do we actually know that they've learned something versus like they know it, but they just can't tell us because they're stressed out? And this becomes a really interesting scenario that we're constantly dealing with. How do we test learning by removing things that are potentially stressful? And so how many of you think that if you had like no stress at all, none whatsoever, that if you were able to demonstrate that you've learned something, that you'd be able to do it 100% efficiently? No stress. Like a little bit of stress now because I'm asking you, so an answer would be good. Do you think you'd be able to do a great job? Yes. If you know the task well. Okay, so if you have no stress whatsoever, you walk into your final exam, you know all the material, you've studied it, would you do really, really well on your final exam? You don't know? I'd hope you would say yes, right? But that becomes a big question. And it turns out that people, when they look at things related to stress and learning, and going back to LTP, one of the things that they're really interested in is how much stress is enough. That's the question that we ask as well in terms of being educators. And the simple answer is that you do actually need stress to help you to learn. Now I'm going to go through this study. I'm going to take you through parts of it. I may not finish all the slides, but I do want to get to the parts that you're going to need for your online quiz. So bear with me if I skip over some slides. But one of the things that's really important is that for animals, we typically give them a physiological stress, a physical stress that mimics your psychological stress. Like you're stressed out all the time by having all these deadlines, by having things that are causing you anxiety. But if we were to take a look, for example, at a physical stressor or a de-stressor, what might that look like? So if we were to take in a Morris water maze, which is this maze here. So this is a wide platform that is filled with water. It's usually opaque. You can't actually see the water, like it's got some colored paint on it. And there's usually a little hidden platform. And the animals are able to find this hidden platform by having some cues that are around the outside. So it learns like, oh, there's that cue over there. If I swim over toward that cue, I'll be able to find this platform. And this is how we monitor their learning. And we can actually track like how long they swim for or how quickly they find this hidden platform when we do this type of task. So one of the things that Lisa Conboy and Carmen Sandy did in a paper in 2010 was to actually see, like, does stress actually impact LTP, learning, as well as memory? And it turns out that if you are given a nice relaxing condition, so you're given a nice warm bath, 30 degrees centigrade is actually fairly comfortable, versus 22 degrees. If you put the animals in 22 degree water and ask them to swim around and find this platform versus 30 degrees, actually the animals that have a little bit of stress, 22 degrees is not something that physiologically impairs them, but they find it uncomfortable. It's below their body temperature. They don't really like it, and it's a little bit colder than they want. They actually learn better. So the time that it takes them to find this platform on day 2, they learn to find that platform really quickly. This is trial 1. They find it even faster in trial 2 on day 2, and by trial 3 on day 2, they find that platform really quickly. Do they remember where that platform is? So if you go back on day 3, they find that platform almost immediately at 22 degrees, and that memory stays with them all the way out to trial 9 on day 4. So a little bit of stress actually helped make their learning and their memory better. Now, if you have a more comfortable condition, it's not that you are not learning. You are learning. It turns out that, again, you still learn well. There's a slope. You learn better every time. It takes you less and less time, but you're not learning as fast. You're not learning as quickly as you would when you have just a little bit of stress on you. And this is what these bars are showing you here, that yes, you are increasing or decreasing the time that it takes to find this platform, but that overall, compared to the counterparts that are in a slightly stressful situation, you are finding it slower. So again, suggesting that a little bit of stress is actually important. Again, they don't have to swim as far, which is what this cumulative distance is showing. The animals in cold water, they don't swim as far. They find that platform right away. Whereas the other animals, maybe they like the water. Maybe they just go for a nice little swim around the water tank before they finally deposit themselves on the hidden platform. I can tell you, having done the Morris water maze, this is really nice, clean data. But once in a while, you have this animal that you, or this mouse or this rat, that really pisses you off, because the data says that these mice don't like being in the water, but some of them really love being in the water. Like, they just don't want to come out. They kind of do a little backstroke. They wave at you when they're kind of doing this little circle. And I can tell you that sometimes the data gets a little bit confounding. Again, if you were to measure, are there levels of stress going up? The glucocorticoid level measured by corticosterone, their stress levels are much higher at 22 degrees than they are at 30. So they are stressed, and that stress is helping them find this platform a little bit better. So again, this goes back to another question that you'll need to know. And this is actually a really good question, and it's some data that I'd like you to make sure that you know for the final exam. So you have to kind of tease apart, and hopefully you'll understand why they're doing these things on this particular graph. So these are control animals not exposed to anything. These are slightly stressed animals at 22 degrees. And in black bars, we have the animals that are at the nice, comfortable 30 degrees. They are measuring GLU-A2 or GLU-R2, same thing, or N-cadherin and NCAM 180. And one of the things that they show is that when they look at the cell surface expression, that after they have learned this particular task of finding that hidden platform under slightly stressful conditions, that the amount of GLU-A2 is significantly higher than the increases that you would see, and they still do see an increase, but the increase is much bigger after a stressful condition. So AMPA receptors are being expressed on the cell surface much better under slightly stressful conditions. But the study is actually a really good one for another reason. These results are specific to the AMPA receptors. They're not looking at the NMDA receptor here, unfortunately, but they are making it specific for the AMPA receptors. Because if they were also looking at this other protein that's found on the postsynaptic cell, N-cadherin is a scaffolding anchor protein that is found, and it is a postsynaptic protein that helps to stabilize only AMPA receptors. If you get more AMPA receptors, you would expect that you would get more of this N-cadherin. And indeed, one of the things that happens is under these stressful conditions, not only do you increase the surface expression of GLU-A2, you also increase the expression of the anchor protein that will hold the GLU-A2 there, or the GLU-R2 there at the synapse, N-cadherin. So is everything just going up? It's not, because if you look at another synaptic scaffolding protein, just a generic scaffolding protein called NCAM 180, under stressful conditions, the synapses aren't just all going nuts. It's only the synapses that contain GLU-A2 that are increasing specifically, because NCAM 180 levels, which is just a generalized synaptic marker, their levels under stressful conditions are not changing significantly at all. So it's a very, very specific phenomenon. Stress affects this LTP-like pathway to increase GLU-A2, therefore AMPA receptors, at the cell surface, exactly as our model would suggest. So ultimately, what does that mean for you as students? It means that you do need a little bit of stress. You might call it motivation, you might call it a deadline, you might call it a final literature review, whatever you call it. You need a little bit of motivation to get you going. If you had like an infinite amount of time, you and I both know that you'll put it off for as long as you possibly can. And these results show that in an animal study, we get exactly the same kind of biochemical data coming back out. So again, if we were to take a look, which one of the following scaffold proteins, which I do want you to make sure that you know, NCAM 180 and N-cadherin are molecules that you're going to see in other courses down the road, and I want you to know which ones are associated with AMPA receptors in the GLU-A2 subunit. So I'm hoping that all of you will remember that it's the N-cadherin that's the really important molecule, that's the scaffolding molecule. So calcineurin and PP1, they might be found postsynaptically, but these are enzymes. These are phosphatases. These aren't ones that are normally associated with a scaffolding function. So again, I'm hoping that will be also really helpful for you for your upcoming online quiz. You'll find that question 4 is indeed very similar to the one that I'm showing you here. So what does this ultimately mean? One of the things that was really clear, you can actually look at the effects of stress. Stress actually is very important. And all of these different effects, the increases in AMPA receptor expression, all of that at cold temperatures, I could go back and I can get everything back to the control level simply by injecting in a blocker for the glucocorticoid receptor, like RU486 that we've talked about previously. So all of these effects that I've just shown you, AMPA going up, the N-cadherin going up, all of that, even at 22 degrees, I could block it by simply injecting in a glucocorticoid receptor. So we don't know how stress actually produces these effects. It does seem to be very consistent in the literature that stress is actually important in your motivation for learning and memory. It might actually be somehow recruiting this CAM kinase 2 alpha pathway that might also increase the levels of BDNF in those neurons. It might actually activate the Krebs pathway, but we're not 100% sure as to how all of these different mechanisms work. The one thing that I will mention to you in passing, and if you're really interested, you can look up this neural plasticity review. If we're looking at the overall levels of stress, the thing that we're doing here at U of T is completely at odds with what we should be doing as educators for all of you, which is really trying to maximize this. We know that all of you have an optimal level of stress that allows you to learn really well. I can't tell like if your level of stress, the threshold is different from someone else. It likely is. But we all know that at some point when the stress gets too high, you're just not going to remember things as well. You're not going to function as well. LTP becomes impaired. And then we enter into this sort of downward spiral. Your memory doesn't work as well. The one thing though that we're really good at is this pathway over here on the left. When we want to scare you, and when we want you to remember something bad that's happened to you, U of T is really good at that. So this fear pathway actually doesn't fall down. It just plateaus. Once you've learned a certain amount of fear, that fear levels stay high no matter how much more fear you add to it or how much more anxiety you add to this process. So I think that one of the things that we should be looking at is how we understand the right amount of stress to maximize your LTP, learning and memory for all students everywhere. So I'm going to eventually get to these two individuals down the road. So I'm going to ask you not to vote on them. We'll come back to vote on these individuals next time. Okay, that's not bad. All right, we'll come back to this in the next lecture because this has been the best sort of story in neuroscience over the last 10 years. In fact, as many of you know, the Society for Neuroscience is holding its annual meeting down in Washington today. It ends tomorrow and all the neuroscientists are coming back. And the two neuroscientists that actually were at this meeting are these two individuals here, and we'll talk about them in the next lecture. So I just need to get through to this one question here. So this one you'll need to know, so I want to make sure that I cover this for you for question 5. Again, apologies on jumping around. So this one question for question 5, I need you to understand a few things. We don't really understand long-term memory. I'm going to try to explain that to you in terms of memory engrams in the next lecture and what we think memory engrams are, memory traces that exist within your brain, sometimes causing false memories to occur, etc. We don't really understand that, although I'm going to try my best to give you the best explanation in 2017. But we do know that the hippocampus is not involved in long-term memories. So things that you remember from 10 years ago, 15 years ago, that's not your hippocampus. That is a different part of your brain, the cortex. We know that the hippocampus only stores memories for about 10 years, and that we also know that long-term memories might also be encoded in another part of the brain near the hippocampus, which is known as the entorhinal cortex, which might be more important in long-term memory storage as well as formation that lasts a lifetime. One of the reasons why we think that is true, if we look at the brains of individuals that have Alzheimer's, which have impaired memories that impact their lifelong learning, they can't remember people from 20 years ago or 10 years ago. The parts of the brains that are severely damaged include the entorhinal cortex. So some suggestion that the entorhinal cortex is important for these really long-term forms of memory. There's also some evidence to suggest that when you are learning something incrementally, so you're learning how to count and you're learning how your alphabet and you're learning in stages, this incremental form of learning also requires an intact entorhinal cortex as well. It's hard for individuals with Alzheimer's to learn something new incrementally because they have damage to this entorhinal cortex. So this gets into this very last question. So what area of the brain has been implicated in incremental learning? Hopefully that's nothing surprising for you. This will be one of the questions that you might see also in your final exam. Can we get that down to 0, please? Thank you. I think, thank you. Oh, okay. All right. So I'm hoping that you guys all will try the online quiz. So this works in your favor in a couple of ways. I can guarantee you that at least one of the questions that you get on the online quiz will be very similar or identical to what you get on the final exam. Number 2, By doing this quiz, we will reduce the impact of the final exam by whatever percent. So this is a 1%. I'm hoping that everyone gets 1 out of 1%. That means your final exam percentage will be reduced by 1%. It's like worth 1% less. You've banked 1% in other words. So you get the questions. You also get your final exam being worth less and less the more of these you do. So if you do all four, which will reduce your final exam mark by 5%, your final exam mark will be out of 30 versus being out of 35. So one other thing before you guys leave. So some of you have been wondering about your final assignment. So let me just mention, sorry, did you guys have any questions about your online quizzes or what the impact is on your final exam? It's always in your favor. If you need to have some of these online quizzes reset, please let me know. I know one student had mentioned that there was an issue where they had logged in. It already gave them one attempt. I can reset those for you. Just let me know. This particular quiz runs out on Sunday. If you can't make it for a variety of reasons, you're out of town or you're doing other things, let me know early. You'll be getting 4 of these in total. Some of them will be worth a little bit more, like 1.5%. Some will be worth a bit less, like this one, which is only worth 1%, but ultimately it'll total up to 5 full percent. I'm hoping all of you get 100% on it. The key is to help you study in advance for the final exam. Yeah, Michael, did you have a question?
So for the quiz, like let's say we don't get the whole 5% for the quiz. So let's say we get like 4 of the 5%. Does that mean that 4% gets taken off of our final exam? Like our exam would be 31%.
So if you tried all 5% worth of questions, then we'll make that final exam, the final total will be reduced by 5%, but the mark that you get out of that 5 will actually count toward your final exam. So if you got 5 out of 5, that means you got perfect on it. If you got like 1 out of 5, that means you've already lost 4%. Don't do that. So if you're having problems with these online quizzes, come and see me. I'll help you make sure that you get 100% on it. Okay? So one last thing. So when you're working through your final literature review, one good suggestion for you, what you're really doing is you are rewriting the introduction. Your paper that you have chosen has an introduction, and you are taking that introduction and rewriting it using other references that you have found. So if you are working on a paper that, for example, looks at the role of beta amyloid in Alzheimer's disease, you will find your own references for Alzheimer's disease, not the ones that they used in their intro. You will find your own references for beta amyloid. Then you will introduce the rationale behind why they did their experiments in that paper. You will then summarize their results from their paper only. You're not doing a literature review of all the different results on that, just that paper. You're summarizing the results. Part of the mark that you're getting on your literature review on that paper is, I don't want to see all of the results. Your TAs have been told if all of the results, every last figure on that paper is on your review, then you're not going to get full marks. You should be picking the results that you feel are going to explain the major findings of that paper the best, writing your own figure legends, etc. So that part is more of a summary of that paper. And then you're going to go through the conclusions that the authors made on that paper. You're going to reword it in your own words and demonstrate that their results match their conclusions. But then you're also going to go back into the literature and see, compared to everything else that other people have found about Alzheimer's or beta amyloid or your topic, how does that fit into the literature? Did they skip some papers that were published? Why did they not put those papers in there? And you're going to start critiquing some of the things that you might find to be improved. Those will be new references that you're going to include. So between the introduction that you're rewriting and the discussion/critique that you're doing, those will probably add up to about 10 new references. You can use some references from the paper that you've been referring back to. So some of those might be overlapping references that you found that they also use in their paper. That's perfectly fine. We just don't want to see the same bibliography that you had in your review paper with the same bibliography that they had in their original paper. Does that make sense? Then you will also suggest What are the future directions? So what should they do next? So you've found some errors, so found some things that they might not have reported on, then you'll make some suggestions as to what to do next, and you might find some other references that are required for you to come up with a good future directions. You might have like 3 or 4 references there. All that kind of adds up to about 15 references. Some of you might have a few more. If you're a few less than that, that's also okay. You should also be contacting your TA or myself if you're having problems with like trying to figure out any part of this assignment. Try to do it as early as you can. I know you have other things. I know you have stress, for example, but you should try to do that as early as you can so that you are reducing the amount of stress that you're putting on yourself, okay? It's a fairly weighty assignment. It's about 20%, or it might be worth 25% for some of you as well. And again, please let me know how we can help. The last thing that I wanted to impart on you, there's a long time between the end of classes and your final exam. I am going to have office hours throughout the entire exam period. You're always welcome to drop in and see me about any questions that you have about the grading, about any of the assignments, about any of the concepts that you've learned here. We will try over the weekend to give you a study guide that covers everything that you'll need to know by the end of the course, including some practice questions. But I also really encourage you to tell me what you need in order to really do well. So have a great weekend. I'll see you all in the next class. Sorry that we ended just a little bit later than usual, but please let me know how to help you the best that I can.